It's a pleasure to welcome Shanika. Shanika has recently submitted her PhD, so huge congratulations to Shanika. This is a wonderful thing. She has an interest in plant pathology, restoration ecology, and environmental conservation. And I really like the idea of Shanika's project because she's looking at rehabilitating Phytophthora cinnamomi infested sites. It's one of these projects of people trying to manage the infested sites, which is so incredibly important. There's so much land that's infested, and there's such a need to improve the management and improve the conservation of these infested sites. Shanika is going to be talking about using pelletized seeds of resistant species within the restoration project. So it sounds like this is a project that could have massive beneficial impacts. So I'm not sure where Shanika is, but Shanika, welcome. Thank you very much.
I'm Shanika from Murdoch University, and today I'm going to talk briefly about my PhD research project, Trade-Based Approach to Rehabilitate Phytophthora cinnamomi infested forest sites. As we all know, Phytophthora cinnamomi is a major key threatening process to Australian biodiversity, and it has caused significant damage to native flora in Western Australia, specifically to the jarrah forest. And these two images are taken from jarrah forest located in around like Shire of Mundaring area. And you can see that in your right side image, the impact pathogen has caused to the forest. So most of the mid and understory species are killed by the pathogen, reducing the species richness and species diversity. And you can see that key susceptible species such as grass trees, semias, and banksias, they are locally extinct. So these changes has altered the structure and composition of the forest, reducing its ability to support its dependent fauna and flora. There are a lot of management practices which we use to reduce the disease spread and mitigate the disease impact. However, still we cannot completely eradicate the pathogen. Therefore, in my research project, I have attempted to rehabilitate Phytophthora infested forest sites with native resistant species. And this project was consisted with three main steps. First, I evaluated the impact of the pathogen in the targeted area, and then I identified the resistant species. And last, I tried to establish the selected resistant species in dieback sites. So as the first step, I evaluated the impact of the pathogen on forest structure, taxonomic and functional diversity in 5 reserves of the jarrah forest located in Shire of Mundaring. So in the map, you can see that these reserves are located close to Perth, around 35 kilometers east of Perth. And these reserves have been previously mapped by the dieback interpreter Joe. And so during the project, we knew that where the infestation was present. So I used the line transect method to collect data. And for forest structure, I collected data on canopy cover and understory cover. And for taxonomic diversity, I used measures such as species richness, abundance, Shannon diversity, and evenness. And for the first time in the jarrah forest, I did a functional diversity analysis to see how pathogen has impacted to the functional aspect of the forest. So for that, I have collected information on 13 plant trade groups, and all the information on these trade groups were collected on 137 species, which I have recorded during the survey. And then these 13 traits were divided into 4 groups, as mentioned here: nutrient acquisition, carbon acquisition, disturbance tolerance, and then reproductive traits. And then for each of these trait groups, I have calculated the functional diversity to see whether there's any impact of the pathogen. So moving into the results, talking about the forest structure, I did not see any impact of the pathogen on canopy cover. However, understory cover was significantly higher in non-infested areas. And for the taxonomic diversity variables, species richness, channel diversity, evenness, all of these variables were significantly higher in non-infested areas, indicating that there is a severe impact of Phytophthora in infested areas. The species abundance was not different between infested and non-infested zones. Talking about the functional diversity, nutrient acquisition and reproductive traits were significantly higher in non-infested areas. And in contrast, carbon acquisition traits were higher in infested areas, and there was no impact of pathogen on disturbance tolerance traits. The diversity was quite similar between in both zones. And talking about the implications for the rehabilitation, the results of this experiment. So I have observed a significant impact of the pathogen on understory cover and most of the taxonomic and functional diversity variables. This indicates that the rehabilitation project needs to include floriferous mid and understory species, which can provide food and refuge to the wildlife. And also, the species which we can use in rehabilitation can be varied on the site-specific impact of Phytophthora. For example, in this project, I have concentrated on mid and understory species because it's the forest layer which pathogen has impacted. But in other cases, it can be the canopy layer. So it depends on the site. And as the second step, I selected resistant species. And for this, I have first selected 50 native species based on the seed availability, seed cost, and also the distribution of these species through southwest of Western Australia. I have also considered other plant traits when I was selecting these 50 species: plant height, drought tolerance, and the ability to provide food and refuge to the wildlife. So I had two experiments. First, I wanted to see how these plants can emerge with the pathogen. So I evaluated the effect of the pathogen on seedling emergence and the survival of these emerged seedlings within first 3 months. So I had sown the seeds of these species in both pathogen infested and non-infested soil. And then I recorded data on seedling emergence. And then I left these seedlings for survival. And after 3 months, I counted the number of survived seedlings. So out of 50 species, I used only 38 species emerged sufficiently to do the stats. And only 9 species, there was no impact of pathogen on both seedling emergence and survival. So this included species from Calathamnus hagia and Melaleuca genus. And the rest of the 29 species, the pathogen impacted either seedling emergence, survival, or both. And in the second experiment, I evaluated when the seedlings, the impact of pathogen when the seedlings are a little bit older. So I have selected 24 species out of previous 38 species. And these 24 species included species which indicated resistance in the experiment 1, and the species which we don't know the disease response yet. And I have also included few species, few non-susceptible species as positive controls. And I have inoculated these plants when they were 5 to 7 months old, and they have left with the pathogen for 2 months. And after 2 months, I assessed shoot growth, root damage, and root dry weight. So using the results I got from both experiment 1 and experiment 2, I have categorized the disease response of these 24 species. So I have identified 2 resistant species in which none of the plants died and pathogen was not isolated from any of the dead, any of the surviving seedlings, and there were no any plant deaths. And then there were 6 mildewed resistant species, and there were 3 tolerant hosts. So in these species, there were no plant deaths, but pathogen was isolated from the roots of surviving plants, indicating that they are hosts. And there were 10 susceptible species and 3 highly susceptible species. So talking about the implications from this experiment for rehabilitation, I have identified 8 resistant species, which I can use these species in the rehabilitation experiment. And the other thing is the species which we think as field resistant based on literature. These species can be susceptible as seedlings, or they can be asymptomatic hosts, which allow the pathogen to survive in their roots without killing the plant. So it's really important to evaluate the disease response of plants as young seedlings and when they mature before we use species in dieback rehabilitation projects. So I have published findings of this experiment in Australasian Plant Pathology Journal. If anyone is interested, can have a read and learn a little bit more about this. The third step is establishing selected resistant species in dieback sites. So for this, I have selected 6 species, 6 native species. So the second species, Acacia saligna, it is a tolerant host which allows pathogen to survive in their roots. And the third species is Banksia sessilis, a non-susceptible species. And the rest of the 4 species, Acacia acuminata, Calathemnus, Hakia, and Melaleuca, those 4 species are resistant to the pathogen. And I included Acacia and Banksia to see how these species perform under field conditions, because we don't know what will happen with these species. And for I have established 18 rehabilitation plots in 3 of the selected reserves, which I did the survey before, and 6 plots in each reserve. And in each rehabilitation plot, I had 6 subplots for each of the species. And in each subplot, I had 24 planting sites for each of the treatments I had. So talking about the treatments, for both Acacias, Calathemnus, and Melaleuca, I had 3 treatments. So in a previous experiment I conducted in the glass house with seed pellets, seeds pelletized with calcium bentonite, diatomaceous earth, and sand, I had promising seedling emergence results. So I wanted to see whether actually these pellets can enhance seedling emergence in the field. And also I wanted to see whether these pellets can protect seeds from seed predators. So I have used pelletized seeds for the field experiment. So I had non-pelleted seeds and then pellets. And also I have included an additive for pellets, rhizobium bacteria for acacias and mycorrhizal fungi spores for Calathemnus and Melaleuca, because they have these associations, and I expected that these associations might provide more benefits for the survival of the emerging plants. And for Banksia and Hakia, I had only 2 treatments because they don't have any specific microbial associations. I had non-pelleted seeds and just pellets. And the number of seeds I used in each of the pellet or control for each species varied based on seed size. So for the seeds, for the species such as Calathemnus and Melaleuca, the seed size is very small. So I had 20 seeds in each control and pellet. And I have buried these non-pelleted control seeds under soil, and these pellets were left on the soil surface. This is how it looks like once I finish setting up the experiment, and these pictures are from the Quell Street Reserve. Before I jump into the results, I just want to briefly mention about the weather during the experiment, because this experiment was based on winter rain and I did not do any of the water provision. So as you can see, after setting up the experiment in winter last year, I've got a good amount of rain in winter and spring, but with the start of summer, there was no rain. And then it kind of started to rain again with the start of the autumn. So here I have shown the seedling emergence result, and I have combined all treatments together. So you can see that Hakia laurina resulted the highest seedling emergence, which was around 88%, followed by Acacia saligna. And majority of the species resulted more than 50% seedling emergence. However, the seedling emergence of the Banksia was very low, and it was around 22%. And when I compare the seedling emergence between treatments, here I have plotted seedling emergence percentage against time. So you can see that in both acacia species, non-pelleted seeds, which is shown in red color, resulted the highest seedling emergence compared to the pellets. And again, in Calathamnus, non-pelleted seeds resulted the highest seedling emergence. However, in Melaleuca, there was no difference of seedling emergence between pellets and non-pelleted seeds. In Hagia and Banksia, the seedling emergence of non-pelleted seeds were higher compared to the pellets. So talking about the survival of these emerged seedlings, I have monitored their survival up to 9 months. And when all treatments combined, the highest seedling survival was obtained in Hagia laurina, and followed by Calathamnus sanguinus. And unfortunately, none of the Melaleuca seriata survived during the summer drought, probably due to their very small seedling size. Talking about the survival comparison between treatments, in both acacias and Calathamnus, non-pelleted seeds resulted higher seedling survival compared to the pellets, but there was no difference of seedling survival between pellets and non-pelleted seeds in Banksia and Hagia. So key takeaways from this project is that glasshouse selection of resistant species made it possible to establish resistant species in dieback sites. And seed pelleting or incorporating beneficial microbes did not provide any benefits on seedling emergence or survival compared to non-pelleted seeds. And this is probably due to early disintegration of the pellets in the field with the heavy rain. And even though I have plated the dying roots of dying seedlings, I did not manage to isolate Phytophthora, and all deaths were probably due to drought and insect predation. And the survival of these seedlings up to 9 months in the field indicated that there is a possibility to rehabilitate Phytophthora infested sites with native resistant species. Just want to thank all of these amazing people who helped me with this project. Thank you.
Just while we're getting the microphones to the speakers, I'd just like to say how impressed I am with the huge amount of work you did setting up that trial and all of those sites. There's a lot went into that, so well done and very fascinating results. And we've got some questions for that.
Yeah, thanks for that presentation. I'm just curious of those 9 native species that were resistant. Are they present at the uninfested parts of?
Sorry?
The 9 species that were disease resistant, are they present at the site in the uninfested areas? And what
No,
frequency?
except for the Banksias, the other species are not present, because most of the present species are like understory shrubs, but there were no any like mid-story species. So my concentration was on mid-story species. So most of these species are not present in uninfested areas.
Any more questions? Tom.
Great project, Shanika. There's a lot of work that went into that. Do you think that seeing that a lot of your seedlings died over the whole summer, would you think you would expect higher success if you're putting saplings in the ground, like more mature plants, rather than going directly in with seedlings?
That could be, but we need to test that. But I think there's a possibility, because if we can plant seedlings very early in the winter, or maybe at the end of autumn, there's a high chance that the seedlings might survive, because they can establish during the winter and then they can withstand the summer. So yeah, it depends. It could be. So there's a possibility to do this research on that. And we are trying to do that as well with the Shire of Mundaring, with Tracy and everyone. So for the next step, we are trying to do that using the seedlings. And also we will be continuing this project for a while to see how these plants survive in the longer term.
We do have plenty of time, so any questions, please feel free to speak up.
Hey, great presentation. Did you do anything to prepare the site, like clear any of the leaf matter or anything before you sowed the seeds? And would you think that keeping leaf matter and stuff would have helped retain moisture or help survival rate?
I removed the leaf litter because I left this pellets on the soil surface, and the seedlings of Melaleuca and Calathemnus are very, very small. And if I left the leaf litter, I won't be able to see the emerging seedlings. So I had to remove the leaf litter. But along the way, I have seen that there's a lot of leaf litter fall as well. So I don't think there would be a very big difference with the leaf litter removal and for the survival.
Any other questions?
Not so much a question, but really, I'm Tracy Bell from the Shire of Mundaring, and so we're really proud and privileged to be able to work with Murdoch in this amazing project. It's groundbreaking, and I just wanted to acknowledge Shanika for the incredible hard work, pretty trying conditions at times. Those field trials were fairly full on, and of course, Giles and Trina and everyone involved. As I said, this work is really exciting because we do have a lot of heavily impacted dieback sites, and we do a lot of work with Joe from Territory, and we've had Bruno help us. So we're really committed to this journey. So we would welcome any feedback, anybody who wants to find out what we did, talk to us, talk to Shanika and Giles. There's a big future in this, it's needed. But we need a lot more investment in this kind of work. This work was funded through the state NRM program, Murdoch and the Shire of Mundaring and others. So we need to keep this going forward, and we would love to see other local government agencies get involved. It can be in a small way, it can be in a big way, but we do, work together and form partnerships and just keep the impetus going, because there's a lot to do, and we're now in discussions about the next phase of the project, and hopefully we can keep Shanika on board a little while longer. We don't want to lose her. So thanks, Shanika. We really appreciate it.
A huge thank to Tracy. Can
I ask you a question?
I've got two questions. Here, there's another one.
My question is just around, and sorry if you covered this, but the cost comparison between the pelletized seed treatment and the non-pelletized treatment?
No, I think there's no very big difference, because the material that we used were like calcium bentonite, diatomaceous earth and sand, so they're not very, very expensive. So if the pellets were successful, because most of the pellets were dissolved in the field, probably due to the heavy rainfall. So I think if the pellets are successful, there mustn't be like huge difference of the cost if we go for a higher scale.
Okay, thank you.
Yeah, thanks, Shanika. I just wanted to add to what Tracy was saying. Some people will be concerned, about not using locally endemic species in the trials, but you got to remember that these reserves are highly modified by dieback. And I guess the idea is, what species can we use that are going to be resistant or have low susceptibility to Phytophthora and can restore some of the lost ecological function with the removal of, species like Banksia sessilis, Banksia grandis. So by using these species, they're not locally endemic, some of them, but they can go a long way to improving ecological function and foraging habitat for birds and small mammals that's lost through the impact of dieback.
No, please, if you have a question.
I was just wondering if, you know, future ideas, whether people would go and sort of collect seed from certain areas and then look at the resistance of what's actually in certain soil types and things like that. I know, I don't know if it's a PACE or a PACE or whatever. They've got sort of soil types and rehabilitation seedlings and things like that that they use. But wouldn't it be wonderful to actually sort of go and collect seed from local areas and see what would survive?
Yeah, it would be really good if we have funding to do that, because we had like limited funding, and this is like 3.5-year project, so we had to, like, we haven't had any chance to do the seed collection, but it would be really good idea to do that, if whether the different provenance can survive more compared to the other. Yeah, it's a really good aspect, which if we can do it, it would be great. Yeah, fingers crossed we will get funding for that.
One more question at the back.
With the insect predation, were they insects identified? Were they native insects or were they introduced?
I think they are native insects, and yeah, I have only seen like few insects, and I think they are native, but not the introduced ones. Yeah.
Wonderful. Thank you very much, Shanika.